Participants, you now have 5 minutes left to the end of the quiz. Participants, there is now 1 minute left to the end of the quiz. Okay, so the 15 minutes are up, but we are giving 5 minutes for those of you who haven't submitted the responses yet. Please submit them as soon as possible.
Okay, so I'm back. And there are some questions. Maybe I should go through the quiz first or answer the questions. Can we use growth factor inhibitors for scarring in order to regenerate CNS neuronal fiber connections? So, yes, it's a combination of all these things, growth factors to cajole, to coax, to persuade the regrowing axon to grow beyond that um, scar tissue, and then inhibitors that blocks those inhibitory factors in the scar tissue. Yes, so it's a combination of all those things that are being tried. It's not going to be one answer. It's going to be a combination of all that. Does MGMT methylation depend on genetic imprinting or varies from individual to another due to some other mechanism? Yes, a good question. There is some genetic imprinting also. There are some epigenetic factors that are transmitted uh, through the through inheritance. We know that also. But in most of the cases, it's a combination that occurs later on as the tumor starts to grow. So, yes, the answer is both, but then more probably it's the um, epigenetic changes later on as the tumor starts to multiply. The cells, they take different genetic makeup and epigenetic modification in each cell. It happens over time. Why do some chemotherapy agents have resistance in certain cancers? Because chemotherapy, just example, temozolomide, it does the alkylation of the um, guanine residues. Now, that is a damage to the DNA, and those kind of damage can occur from environmental factors also. So our body has mechanism to protect that, MGMT, for example. So the MGMT methylation can counter the effect of chemotherapy. MGMT methylation silences that, and if MGMT is not methylated, then MGMT is produced, and that counters the effect of temozolomide in particular. So there are different mechanisms for different chemotherapeutic agents, and that's why they develop resistance. As the cells proliferate, they have a lot of genetic and epigenetic variations, and those are selected which are resistant to chemotherapy, and they keep on multiplying. So that's why the idea of precision medicine is coming in, that you can't treat all the tumor cells by one mean. You have to modify, and the idea is that you can take samples from each, and it is being done in other cancers also. The genetic and the molecular analysis of those tumor cells is being done, and then precise medication is tried so that the resistance is minimal to that chemotherapeutic agent. So that is a hot area nowadays. Why do you think advertisement of stem cell therapy isn't true? What is biggest barrier to it? Uh, why do you think advertisement of stem cell therapy isn't true? So a lot of these advertisements on stem cell therapy, they are not based on good science, not based on good clinical trials. They are just there to um, uh, get patients and fool them by saying, we will treat this thing. There is no data that has shown, for example, in spinal cord injury, that a stem cell transplantation has helped those patients. there are anecdotes here and there, but then that can be anything. So if I have a spinal cord injury patient, may not be able to walk initially, but then over time, some of those patients may start to heal and may start to walk. So if you did something in that patient, for example, you may say, "Oh, because I did this, that's why I started to walk." How will we clear this up? Is by statistical analysis, by doing a randomized control trial. So these stem cell therapy advertisement that we have, they are very misleading. And what is the biggest barrier to it? So I feel to understand that biggest barrier to the advertisement or biggest barrier to stem cell therapy. If it is biggest barrier to the advertisement, then that's the issue of ethics. That's the issue of our morality. If the biggest barrier to stem cell therapy, then yes, the biggest barrier is to understand its physiology and to understand how to control them. That is the biggest barrier. Okay, GBM is highly invasive, which makes total gross section hard. So what are some future potential treatments? So the potential future treatments would be that those cells that are still in there, how can you control those cells? How can you differentiate them and let them sit there? So again, a lot of factors one can use. One can use gene therapy to modify those cells. One can use brachytherapy to kill some of those cells. One can use precision medicine, maybe even microRNA sponges, potentially, can infuse in that area, and it will bind and get endocytosed by those tumor cells and stop their cycle. The whole mechanism is so complex, as I showed you in the apoptotic pathway. There's so many factors that are affecting, and each one of them is affected by so many things, like I showed in the AKT. So it's very difficult to get there, but potentially, theoretically, if we control all that thing, we should be able to control those cells. Okay, so can I have the the link to the quiz, please? Yes, thank you. So let's see. All right. Share screen. And, okay. So, so in this regeneration in peripheral nervous system is more successful than in central nervous system, mainly because inhibitory factors in peripheral nervous system. There might be some inhibitory factors, but we don't know. Inhibitory factors in the central nervous system, that's what we talked about, and here is the diagram also. Difference in genes of neurons in peripheral nervous system. So remember, those neurons, most of them are already in the central nervous system. And I showed an example that genetically the motor neuron can bypass through another graft. So it's nothing wrong with that. It is incapable of regenerating. There's not a difference in the genes. They all have the same genes. And cerebrospinal fluid, obviously, it does not seem to have an effect on central nervous system or peripheral nervous system regrowth. All of the following have shown to contribute to inhibition of regeneration. Myelin-associated glycoprotein, yes, we talked about it, and actually it's right over here. No-go receptor, chondroitin sulfate protoglycan, all these three over there. The epidermal growth factor, so far it does not look like it's affecting regeneration. It has a role in tumor growth. Stem cell therapy for neurological diseases have been proven and are being offered with remarkable success. No. Have no role and should not be considered. That's not true either. They may have a lot of role and should be considered. Have not shown any benefit in any clinical trials in any disease. That's not true. There are a lot of clinical trials that are being done in phase 1, phase 2 on different diseases, ALS, multiple sclerosis, traumatic brain injury, mesenchymal stem cell therapy is being done and has shown some effect. And so the answer is preliminary clinical trials have shown some benefit in some diseases. Induced pluripotent stem cells are created by deleting specific genes in the cells. No, that's not true. Adding specific transcription factors. Remember um, that experiment that we showed of what was his name, uh, Yamanaka, which showed that those 4 transcription factors. So it's not methylating growth factors, it's not removing differentiated cells, it's adding specific transcription factors. At this point, would reprogram the fibroblast into the induced pluripotent stem cells. Stem cell therapy has a role in ALS, yes, MS, yes, spinal cord degeneration, yes, all of the above, yes. So that's the answer. Brain tumor stem cells, glioma. Brain tumor, so the question should be brain tumor stem cells in glioma limit the growth of glioma. No, they actually are the source of growth of glioma. And they're not identical to the rest of this. So this one is different than this one. This may be CD133 or CD44 positive. These are CD133 and CD44 negative. They die early during a phase of tumor growth. That's not true. So the answer is are the source of growth of glioma. Converted proteoglycan, this one, is a cell surface molecule. No, it's an ECM molecule. It's released out. It inhibits regeneration. Yes, it can. It promotes tumor invasion. No, it actually inhibits tumor invasion also. I showed actually one of my experiments there. It's a transcription factor. It's not a transcription factor, it's an ECM protein. So the answer is it inhibits regeneration. The 3 main pathways that may be involved in glioma invasion are following except. So actually I did put the picture in there. So one of them is sonic hedgehog pathway, which is this one, sonic hedgehog and TGLI1. Wnt beta-catenin pathway, which is this one. And PI3K-AKT pathway, which is this one. And then SOX2-MEK. These are the 2 transcription factors that are used in iPSC. So that's the wrong answer. That's the answer. Except. So except this one. Following is true for temozolomide, except. I had a lot of questions with except. Sorry about that. It's an alkylating agent. It adds methyl residue. It adds methyl to guanine in DNA. That's true. MGMT protects cells from it because whatever it does, MGMT comes and takes it away, fixes that. So the right answer, except that it inhibits p53. So it has not been shown that it affects p53 directly. p53 is a tumor suppressor gene. MicroRNA are non-coding RNA. They don't code. They are similar to tRNA. They are different than tRNA. They are attached to ribosomes. No, they're not attached to ribosomes. They're in cytosol and they're exocytosed. And they are released from cell. So the right answer is they're non-coding RNA. Following are stem cell markers in glioma initiating cells. So this is a little tough, but we talked about LRG5, CD133, CD44. These are all tumor stem cell markers. IDH1 is an enzyme. It's not a cell surface marker. It's an enzyme inside the mitochondria in the cytosol, which is important in turning isocitrate into alpha-ketoglutarate. So that is the answer. Mesenchymal stem cells have shown beneficial effect in most of these stem cell therapy trials and experiments because they generate exact cell relative to these stems at the site of injection. So far it looks like that these mesenchymal stem cells, they don't differentiate into those kind of cells where it was transplanted. They release protein, peptides, and RNA which promote health of surrounding cells. So that's what looks like the answer. They are totipotent in their potential. They're not totipotent. They are further differentiated, but they are stem cells. They can still differentiate into some more cells. They replace all the damaged stem cells in surrounding. That's not true. They should stay there. Rest of the structures start to have develop some function. So the right answer is this one. Okay, so we did that. Now there are some more questions that have come in. Can extracellular matrix components be injected in an individual's brain to halt the progression of brain atrophy? So let's say if you inject it, atrophy is throughout the brain. If you inject it over here, you will just inject it over there. It's not going to diffuse out, number one. Number two, ECM what component of ECM you think will halt the progression of brain atrophy? So brain atrophy doesn't occur because of lack of ECM. Brain atrophy occurs because of a lot of other factors. Remember, in our talk yesterday, we talked about how amyloid precursor proteins um, um, uh, modification in a wrong way would lead to amyloid plaques, how alpha-synuclein aggregates are killing cells, and making Lewy bodies, how aggregates of other proteins uh, were damaging cells. So those are the factors. So ECM, I don't know if ECM can really do the trick. I don't think so it can do the trick. But certain ECM components can be inhibitory, and if you can control the expression of those, then yes. In a developing brain, the ECM is very loose because there's a lot of hyaluronan present, hyaluronic acid. Those are big molecules which creates a lot of space, and the neurites can go through those spaces better. So that is good in embryogenesis, but then to prevent atrophy, I don't think it's going to help. How do patients recover if there's no regeneration in CNS? So if we're talking about, let's say we're talking about let's take three examples. So this spinal cord was injured. The patient went through shock. There was some damage. Some axons were totally damaged, and they would not regrow. But then in the periphery, there were some axons which went through partial damage. Maybe the myelination was affected a little bit, but not totally gone. Though they would recover in a similar way. For example, if there is a pituitary adenoma, which is pushing on the optic nerves, the patient is getting blind. You take away the pituitary adenoma, the pressure is gone. Some of those fibers, they do not recover. Some of them recover because they were not totally damaged. So that's how the recovery takes place. If it's, for example, a stroke, let's say I get a stroke right here in the premotor area, which is controlling my motor area. So I will certainly get paralysis on the left side. But then remember, we talked about neurodarwinism, we talked about synaptic plasticity. So other parts of the brain will start to develop some circuitry that would take over the control that function based on the plasticity that I go through physiotherapy and all that thing. Those areas may then start to make connections with the motor cortex and pick that up. So, but if it's a primary motor cortex that's damaged, then that does not get better. So synaptic plasticity is also a part of this recovery in the damage to the CNS. So it's a combination of different things that causes that. Okay, so those were all the questions that I had. Do you want me to go over this? I could go over this lecture quickly again. So, all right. Okay, so quickly going over the lecture. So we talked about the regeneration in the peripheral nervous system, how after regeneration axon starts to grow, and the Schwann cells and other cells around it make a scaffold, and then the myelin is reproduced. But then in the central nervous system, the damage seems to be permanent, and it turns out that the astroglial scar in the central nervous system is one of the factors that affects it. And in spinal cord damage that occurs, these astroglial scars and the other immune cells and physical cyst formation and lack of myelination, inhibition of the growth of axons, all of this combination affects regeneration in the spinal cord, for example. And in this experiment, when a central nervous system tissue was transplanted, which is optic nerve, there was no growth in the peripheral nervous system. And if a peripheral nervous nerve was transplanted in the cut optic nerve, it regrew there, suggesting that there are inhibitory factors in the central nervous system. And here, a sciatic nerve or a peripheral nerve, when was used to bypass the scar, there was growth through that. So it turns out that there are a lot of inhibitory factors, but then some of them are very well known. That's NOVO-A, oligonucleotide myelin glycoprotein, myelin-associated glycoprotein, gonadotrophic proteoglycan, and their receptors may be involved in this inhibitory process. And not only that, one can modulate these these neurons by not expressing the receptors and not inhibited at those sites. And that makes the basis of a multi-pronged approach to achieve regeneration. Hasn't been achieved yet, but hopefully, if it's done correctly, eventually we will get there. And by the way, this whole thing about brain transplant and all that thing, it's not. It's something that theoretically is possible, but think about it. If you can't even get a simple spinal cord regenerate at a lower level, how can you transplant the whole brain? But there are some experiments done. I don't know about the validity of all those experiments, but yes, theoretically it's possible. And then looking at some of the lower invertebrates where the regeneration takes place can give us a lot of clues how to help regeneration in higher mammals. Okay, and then we talked about the stem cells and how the hoax has really covered that field, but there's a lot of hope in there. Initially, however, in the Parkinson's disease, it was not very helpful, but recently a lot of stem cells from different areas, particularly mesenchymal stem cells, are being used in clinical trials. And the idea of reprogrammed induced pluripotent stem cells got Nobel Prize for Yamanaka, and just the 4 transcription factors he used them to regenerate the induced pluripotent stem cell from the fibroblast. And not only that, the microRNA can also control that. So that is an area that is being studied and looked at in detail. And these stem cells, either differentiated or induced, can be used to produce theoretically, replace whatever is damaged in the nervous system, theoretically. And here's an example of amyotrophic lateral sclerosis, that how you can take a fibroblast and bring it back to induce pluripotent stem cell and then differentiate into motor neurons. Fibroblast stages into motor neurons. Theoretically possible, in vitro possible, in vivo has not been done yet. But there are some other things that are being done in clinical trials, such as in multiple sclerosis, these oligonucleotide progenitors are being injected and that they may remyelinate things. And there are some experiments going on for the transplantation in Parkinson's disease. But then the whole field is fraught with all these fake and deceptive messages that we have to be careful about. And we have to be careful about, if somebody tells us that something has happened, it doesn't mean it has happened. There may be a lot of explanations. I think it was Carl Sagan who said that, I mean, I don't remember the saying, but then the more, the chance of something happening that has happened is well, I don't remember saying it, but he said that the rarity of something that happened, it requires more proof. So if I say that, I found a dead person walking, so I need a lot of proof to convince that. If I say that I found my cousin walking in my home, that's very possible, right? Maybe he was visiting. So that's what I probably didn't say it very eloquently. Anyway, okay. So Then I came about the tumors, and we talked about that how these cells are infiltrating all around here, um, and how there are different kinds of cells, different kind of classes of these highest, most malignant brain tumor, GBM, there's classical, mesenchymal, neural, and proneural, and they all have different genetic modifications and expression of different genes. And there may be genetic disruption also in those cells. So their genetic makeup might have changed also. There might be some deletion, some multiplication of certain gene and all that takes place because it becomes very unstable genetic profile of those cells. But then all of them do arise from the brain tumor stem cell, and the brain tumor stem cells, these are some of the known markers. They are the ones that give rise to brain tumor. And if we can control these brain tumor stem cells, then probably we can control the brain tumors. Some of the factors on the cell surface and some of the release factors may be involved in these brain tumors invading invasiveness. And that's why I say neoplastic invasion recapitulates cell migration during embryogenesis. Fibronectin, I gave an example. Protoglycan, I gave an example how it accumulates and inhibits tumor growth. And the metastatic so maybe we can study this, that how come a metastasis doesn't invade. So an analysis, understanding of this would help us understand how to prevent invasion by the primary brain tumor such as glioma. And some of the experiments I showed. This was just a diagram to show that how complex the apoptotic pathway is. It's not just BCL2 and BAX, yin and yang that we see and read about. It's much more complex than that. And there are a lot of factors that are affecting this, and some of these factors are the same factors that were involved in embryogenesis that we talked about. So some of the known pathways that people are working on, and some of the known chemicals, known proteins, RAS, MAPK, PI3K, P10, P53, CDK4, and all those are being worked on. They have different effects on different things, and some of these are important in migration and invasion. But then it's much more complex than that simple diagram. Even in AKT, in this diagram, in this schematic picture, shows there's so many factors affecting that. So I showed in a video that the main factor that caused that put a stem cell on that pathway, that factor may be gone. We will not be able to detect it, but you see all the pileup from that factor change, and you are studying all those pathways and all those things, but those are as a secondary phenomena. So looks like that the three pathways that we talked about, focusing on these may yield better results. Then I talked about the temozolomide as an alkylating agent, methylation of the guanine residues, and how MGMT protects the cells, even the tumor cells, against the temozolomide. But then the methylation of MGMT silences it, and it doesn't express that. So TMZ have more effect on those tumors which have methylated MGMT. Then I talked about the IDH, how the IDH mutation leads to high 2HG, which has a genetic effect over here, and the role of IDH in tumorigenesis, the wild type versus the mutated type. And TERT is also one of the enzymes that is being studied. TERT is part of telomerase, and telomerase, functional telomerase was mentioned over here. And then in general, there are oncogenes and there are tumor suppressor genes. So EGFR is a typical promoter. It promotes, and P53 is a typical suppressor. But there are a lot of other things like 1p19 Q code deletion in oligonucleotide. How does it work? We don't know. And then in the end, we talked about the microRNA, that there's a new whole area about microRNA that has come up in the last decade to 2 decades. And working on this area probably will give us a lot of answers in tumor growth and a lot of other things also. So that's what it was all about. And I think we can stop sharing over here. I seem to have lost it. Okay. Let's see. Goodness. No, it's not working. Okay. Okay, all right. There it is. So, anything else? It's 11 o'clock. I guess we meet next weekend, which will be the last module. Then there's one lecture after that. And the last module is, there's not much stress in that module for those who are participating, because it's just a general discussion about different things. But there has to be quiz, unfortunately. Okay, so until then, I wish you all peace and Assalamu alaikum and Khuda Hafiz.